Okay, so we were discussing about the gram positive gram negative cell wall. I have explained you about the structures of these two membranes. Like peptidoglycan is a major component of cell wall. For gram negative bacterial cell wall, I told you it also have the outer membrane, and the outer membrane of a bacteria of this gram negative bacteria, it contain an endotoxin. Endotoxin is actually a compound which it's a chemical which produce inflammation in the body. So the gram negative cell wall, the gram negative bacteria, its outer membrane, the membrane which is outer than, which is the outermost membrane, that actually produce endotoxin. And remember this, the outermost membrane of a gram negative bacteria is also used as an identification in labs. The polysaccharides and the proteins which are present in the outermost membrane, which are present in the outermost membrane of gram negative bacteria, it acts as a antigen. You know what is antigen? What is antigen? You haven't studied immunology, you are studying nowadays, right?
Yeah.
Okay. So antigen is actually, antigen mean that that component of a cell which can provoke immune response in the body, that component of a cell or of a tissue is known as antigen. So the polysaccharides and the proteins which are present in the outermost membrane, it also act as a antigen. And this, and from this antigen, we can identify the bacteria in the lab. For example, for this Vibrio cholera, Vibrio cholera or typhoid. Let's suppose discuss about typhoid. For typhoid, we have O antigen, that is O antigen. It is a diagnostic criteria, lab criteria for enteric typhoid bacteria, Salmonella typhi. Salmonella typhi, it's a gram negative bacteria which cause typhoid. You know what is typhoid? Typhoid is also known as enteric fever. So Salmonella typhi is the name of bacteria which cause this disease. And typhoid bacteria, it contains an O antigen that is present, that is that is also known as a somatic antigen, which is present on outer membrane. And this O antigen is uh, is responsible, not responsible, you can say it is a diagnostic or a identification point for this bacteria. So in the lab, we can identify this O antigen, and then we can say that the patient have typhoid fever or a Salmonella typhi infection. Along with that, the outer membrane of a gram negative bacteria, it also contain a small channels. Channels mean a small holes, a small channels that are known as porine. That are known as porine. Sorry, porine channels. So through these porine channels, for example, I think here this is, so through these porine channels, these are the small spaces. Through these porine channels, the hydrophilic compounds, Which one? What do you mean by hydrophilic?
which do not fear
Hydrophilic.
water.
Water soluble.
Water
Water
soluble.
soluble.
Water soluble hydrophilic compounds, like we have some sugars, we have some vitamins, amino acids, some metal ions, or some water soluble compounds, because we know that the the polysaccharides, lipopolysaccharide, they are they are lipid in nature, so they are hydrophobic in nature. So the cell wall of a bacteria, it is hydrophobic. It means it repels water. So that is why the water soluble components cannot cross the membranes. So that is why the outer membrane of a gram negative bacteria, it has some channels, small holes or pores that are known as porine. And through this porine, a water soluble components can enter into the cell. So certain sugar molecules, protein, amino acids, vitamins, metal ions, or water soluble nutrients can can enter into the cell through these channels, and these are known as porine. And these porins are present only in gram negative bacteria, because outer membrane is present in gram negative bacteria. And through this porine channel, the antibiotics like penicillins and cephalosporins can also cross the outer membrane and enter into the cell wall. And when the antibiotic enter into this periplasmic space, so periplasmic space contain beta lactamase enzyme, so which degrade the beta lactam antibiotic. Class understood?
Yes,
Yes,
sir.
sir.
So this is actually the composition of a gram negative bacterial cell wall. So gram negative bacterial cell wall, it has a outer membrane, and outer membrane, it contain endotoxin. Endotoxin is a compound, is a chemical due to which our body produce a immune response, exaggerated immune response, and that will lead to a shock. That is known lead to a shock, and this type of shock is known as septic shock. This is a very important emergency of a gram negative bacteria, septic shock. The patient in a septic shock can like uh, the death occur in those patients if it is not treated urgently. So only gram negative bacteria, remember, only gram negative bacteria, they can cause septic shock. They can cause septic shock because because the lipopolysaccharide which are present outside, which are present at the outer membrane, it contain endotoxin, which contain endotoxin. So these endotoxin produce a immune response that lead to a shock. So patient will develop hypotension and loss of consciousness. And due to hypotension, patient may die. So the bacteria, it produce two type of toxin. One is known as endotoxin and one is known as exotoxin. Endo mean inside, exo mean outside. So endotoxin, why we call this endotoxin? Because the compound, it remain attached with the lipopolysaccharide. So it is produced by the bacteria, but it remain within the outer membrane. So when bacteria enter into the body, enter into the blood, immune response is generated against this endotoxin. While some bacteria, they secrete the endotoxin. They secrete the toxin, and that secreted toxin will present in the blood. For example, have you heard this tetanus?
Yes,
Have you
sir.
heard this tetanus?
बोलिए.
Yes.
So,
Yes, sir.
Yes, sir.
okay, so
Molvi.
tetanus is not small v. Okay, so tetanus is caused by
Molvi.
tetanus is caused by Clostridium tetani. Clostridium
Tetani.
tetani is a bacterial name which cause tetanus. So this Clostridium tetani, it secrete a exotoxin. It secrete a toxin which release in the blood, and within the blood this toxin it circulate. So
What's the name again?
which bacterial? Clostridium tetani. Clostridium tetani. So this bacteria, it produce exotoxin that circulate in the blood, and that will cause a tetanus. It's a neurotoxin. It's a neurotoxin because it block the inhibitory neurotransmitters. So these, while the gram-negative bacteria, the lipopolysaccharide, it contains a endotoxin which remain with the lipopolysaccharide, or which remain within the outer membrane. So that is why we call the endotoxin, because it is not released into the blood. So that's why we call it endotoxin. So let's discuss the peptidoglycan. So peptidoglycan, as I told you, the name suggests, the peptidoglycan mean peptides and sugar. There are two other names of peptidoglycan unit. We call it murin and mucopeptide. Murin and mucopeptide, these are also the term used for peptidoglycan. Murin
And
or
mucopeptide.
mucopeptide. These are also the synonyms, or you can say that these are also the same word which used for peptidoglycan. So peptidoglycan, it is made up of two units, major units, repeated units. One is N-acetyl glucosamine and N-acetyl muramic acid. N-acetyl glucosamine and N-acetyl muramic acid. Muramic acid, there is a mistake here. It's muramic acid. N-acetyl glucosamine and N-acetyl muramic acid. These are the two major component. It's a carbohydrate backbone, because N-acetyl muramic acid and N-acetyl glucosamine, they are actually the glucose unit. These are the glycan units, and each this muramic acid is this black one. Here you can see this, and each muramic acid, there is a attachment of peptide chain. Actually, this is tetrapeptide chain. Tetrapeptide chain. Here you can see each N-acetyl muramic acid, we have 1, 2, 3, 4 peptide linkage, or 4 peptide or amino acids. For example, and these amino acids, the the amino acids, they vary bacteria to bacteria, species to species. Not all bacteria have the same amino acid. For example, Staphylococcus aureus, they have alanine, L-alanine, D-glutamine, lysine, and alanine. While E. coli, it has alanine, glutamine, and instead of lysine, they have another amino acid, that is diamino pymalic acid. Instead of lysine, they have this DAP molecule. So the unit, the peptide units or the amino acid, they vary species to species, even bacteria to type of bacteria. Different type of bacteria, different type of amino acids, they are present, they are attached with this N-acetyl muramic acid. Class, any question? Do you understand?
Yes, Lavashri.
So N-acetyl muramic acid, there is an attachment of tetrapeptides, because four amino acid, they are attached, and the amino acid, the type of amino acid, it differs between species as well as between bacteria. So different bacteria, they have different type of amino acid. So the most important one amino acid, the worthy one, is diamino pymalic acid. It is a unique amino acid which is only present in bacterial cell wall in nature. Okay, remember this. Diamino pymalic acid, or a DAP molecule, it is a only amino acid which is present in bacterial cell wall. It is very unique. On the other hand, the D-alanine, this D-alanine, this one, D-alanine, in picture, this one. So D-alanine is responsible for cross linkage between two peptidoglycan chain. Can you see here? What is this? This is D-alanine. So this D-alanine is cross linkage. It is actually making a bond with another amino acid which is present on the second chain of peptidoglycan. So this is known as peptidoglycine bridge. So peptidoglycine bridge, it is actually because of this D-alanine. So D-alanine is very important, as well as this DAP molecule, because DAP molecule is only unique. It is only present in bacterial cell wall. While for the cross linkage, D-alanine is very important. Okay. So D-alanine is required. It must be there for the cross linkage. Is okay?
Yes, sir.
Okay. And remember this, the D form, this one, the D-alanine is not abundant in nature. Have you studied this classification of amino acid and classification of carbohydrates? I think in biochemistry you must have studied.
Yeah, we
Yes.
did.
Okay, so which form of carbohydrates are abundant in nature? L form or D form?
D form.
Very good, D form. The carbohydrates, the naturally occurring carbohydrates, they are dextrorotatory or D form, while the L form is more abundant, is more common with amino acid. So in nature, the levorotatory form or the L form of amino acids, they are abundant. So this D-alanine, it's also a rare form of amino acid, and it is also unique to bacterial cell wall. Okay, so that's why I mentioned these two things. One is DAB and the D-alanine. So the cross linkage is very important. The cross linkage is very important. So the cross linkage is between this D-alanine and any other molecule of any other peptide or amino acid of second chain. Between this between this linkage, amino acid molecules, they are also present. They are also present, but it also vary bacteria to bacteria. For example, in Staphylococcus aureus, the Staphylococcus aureus or Staph aureus, if you see here, the cross linkage or the peptidoglycine linkage, the 5 amino acid molecules are present. This is also unique to Staphylococcus aureus. The glycine bridge between two, the glycine bridge between two peptidoglycan chain, the cross linkage between the cross linkage is made up of 5 amino acid molecule. This is unique to Staphylococcus aureus. So different bacteria, they have different this glycine link. Class understood?
Yes, sir.
Different bacteria,
Sir.
they have either they have one
Different
amino acid, or either they don't have any amino acid between their linkage. Here you can see in E. coli, there is no any peptide or any amino acid present between this cross linkage or glycine linkage. While in Staphylococcus aureus, 5 amino acid molecule, they are present in glycine bridge or glycine linkage. So different species, different bacteria, different number of amino acid which are present in glycine bridge. Another important thing is here, this glycine bridge, because cell wall is only present in bacteria. Cell wall is only present in bacteria, not in human cell. So that is why when we give penicillin, cephalosporin, they only inhibit the synthesis of peptidoglycan in bacteria, not in human, because human cells, they does not have any cell wall. So that is why they have a limited toxicity. That is why they does not cause any damage to the human cell. So the exact mechanism of action, now I can explain that. The exact mechanism of action of the penicillin and the cephalosporin is they block the enzyme that is known as tetrapeptidase. They actually inhibit the, sorry, not tetra, it's transpeptidase, sorry. Transpeptidase. So transpeptidase is an enzyme which is responsible for this D-alanine cross linkage. Transpeptidase is an enzyme which help this glycine bridge, which produce this glycine bridge. So penicillin and cephalosporin antibiotic, they block this transpeptidase enzyme. When you block this transpeptidase enzyme, there will be no linkage form. So when there is no linkage form, the two peptidoglycan chain cannot join together, because the joining of two peptide chain, it is because of this glycine bridge. So when you inhibit this transpeptidase enzyme, there will be no cross linkage. So there will be no you can say attachment of one peptidoglycan chain with another. So what will happen? The bacteria will lose the cell wall integrity. When the bacteria loss, yeah, it will be disintegrated or integrity will be lost. So when cell wall is lost, when the cell wall is lost, what will happen?
Osmotic blast.
Yes, very good. When you remove the cell wall, the water enter into the cell and it will cause the bursting of cell. This is the mechanism of action of these cell wall synthesis inhibitor drugs. Okay? Because cell wall, it give a strength to the bacterial cell. There is another enzyme, natural enzyme, that is called lysozyme. Lysozyme, it's actually a enzyme which is naturally present in human tears. It is naturally present in human tears, the mucus and the saliva.
Sir,
The
what is found?
lysozyme, yes,
Is found.
yes. You have any
Sir,
question?
I lost your voice in just now. I will disconnect.
Okay, okay. So the lysozyme, it is an enzyme which is present in human tears, mucosa, the mucous membrane, and the saliva. And this lysozyme, it has ability to break down this N-acetyl glucosamine and N-acetyl muramic acid bond. Okay, so this actually the enzyme which break down this glucose backbone of peptidoglycan. So that is why the tears, the human tears, they are natural antibiotic. The saliva is a natural antibiotic because it break down the linkage between the peptidoglycan units. So that is a natural antibiotic for human, and it is a natural resistance against the infection. Okay, class, any question?
Mayo.
Okay, so here you can see the same. We have this sugar backbone, N-acetyl muramic acid and N-acetyl glucosamine, and these are the cross linkage. These are the cross linkage which have tetrapeptide like 1, 2, 3, 4 tetrapeptide, and this is D-alanine which produce a cross linkage or a bridge, glycine bridge, and this glycine bridge, it varies species to species. Some species have 5 amino acid in this bridge, some species have 2, some species have nothing, no bridge, no amino acid. Okay, so next we have teichoic acid. Teichoic acid, these are actually the fibers, and this teichoic acid, they only present only present in gram positive bacteria. They are not present in gram negative bacteria.
Negative.
Yes. So these fibers, they are protruded outside from the cell wall of a gram positive bacteria, and these fibers, they are composed of glycerol phosphate or ribitol phosphate. Their main chemical composition is glycerol phosphate or ribitol phosphate. Some of the teichoic acid, some of these fibers, because they are present on the surface of cells, they are present on the surface, but some of the fibers, they penetrated and attach with the cell membrane or a cytoplasmic membrane with a covalent bond, and this type of and these fibers, they are known as lipoteichoic acids. Some fibers, they penetrate and they are covalently linked to the lipid molecule in the cytoplasmic membrane, and they are called lipoteichoic acid. Okay, here this is teichoic acid. They are protruded outward from the outer membrane, and some of the teichoic acid, they are covalently bound with the cytoplasmic membrane, the lipid of the cytoplasmic membrane, and these fibers are known as lipoteichoic acid. These fibers, they are known as lipoteichoic acid. Class?
Yes, sir.
Is okay?
Yes, yes, sir.
Okay, so the main clinical significance for this teichoic acid is its pathogenicity. Just like endotoxin of gram-negative bacteria can cause an immune response, the same teichoic acid, it produces inflammation and shock due to gram-positive bacteria. And this teichoic acid, it also helps to identify in a lab, because this teichoic acid, it acts as an antigen, and it is recognized by our immune system, and our immune system produce antibodies against this bacteria. So teichoic acid is a pathogenic or a virulent factor. It is also known as a virulent factor.
Viru.
Yes, virulent factor. What is virulent factor? Which produce virulence. Virulence means disease. So teichoic acid, it is just like a endotoxin of a gram-negative bacteria. Okay, class, it is enough for today. I need to take your attendance because we have about 4 minutes. Please be quick. Where is your attendance?
मौलवी
आ गया तू?
Today is 3.
आ गया तो जवान होके
मौलवी तुम मस्ती कर रहे हैं.
Okay, please, your attendance 802. Be quick.
Present. Present.
Okay, 803. 803. 804. 804. 806.
Present.
808. Please
Present.
be quick.
Present.
9. Okay, 9.
Present. Present.
10. 10.
Present.
Okay, 13.
Present.
14. 14,
Present.
17,
Present, sir.
18,
Present.
19,
Present, sir.